Hi, I'm Ushma Neal with the Journal of Clinical Investigation. We are lucky that Dr. Christine Seidman of the Harvard Medical School joins us today for the next in our series of conversations with giants in medicine. Dr. Seidman, also known as Cricket, has uncovered the genetic basis of many human cardiovascular disorders, from cardiomyopathy and heart failure to congenital heart malformations. Today we hope to hear about everything from her early intrigue with atrial natriuretic peptide to her more current gene-intensive investigations. So welcome.
Thank you.
Can you start by telling us where you grew up and about your parents and about your early interest in science?
Certainly. I was born in Kalamazoo, Michigan, but early on I moved because my father was transferred to New York and grew up on Long Island. Subsequently went to high school there and went on to college at Harvard School and then went on to medical school at George Washington in DC. Moved back north to Baltimore to do internship and residency at Johns Hopkins and subsequently cardiology fellowship at the Mass General Hospital.
So I read in an interview that you gave once that your interest in science was even from an early age, and that you almost put yourself out trying to concoct anesthetics to anesthetize butterflies and insects you caught in your garden.
Sadly, that's true. I was very interested in science as a child and in growing up, but I didn't have any exposure to people doing science at a professional level. In fact, the closest I came to seeing individuals who were really devoted to science as career were physicians. We had a general practitioner who would come to our home and visit when one of us was ill, and I think that certainly was the first connection that I made that suggested that science might not just be about bugs and butterflies and things I could dissect under a microscope, but could be actually about living people.
So did you decide early on that you wanted to focus on science or being a physician?
I think it was probably a little bit of both. Again, having no direct exposure to people doing scientific investigation, I early on assumed that science and medicine were one and the same. And I actually think that's still true. I think it's the foundation of good medicine. Good science is good medicine, and I think that that's something that's driven most of my interest in those two disciplines throughout life.
So when you were doing your residency at Hopkins, you were there at the same time, and I've read that you chose Victor McKusick as one of your early inspirations.
Yes.
Did he turn you on to genetics, or were you already sort of on that path towards finding genetics to be fascinating?
No, I certainly was not even aware of what genetics was or how it could be applicable to human disease or basic scientific discovery in those years. Victor McKusick was an incredible individual, a brilliant scientist, and an amazing mentor. He took the chance of taking people who had come through traditional medical approaches and saying, maybe you should think about science. And the approach that he took was very personal. He invited us to his home. We would be well fed, something quite remarkable during internship and residency by his good wife, Anne. And then we would discuss a scientific manuscript. And those discussions were lively, they were interactive, they were not stilted or anything threatening, and one could ask questions and really begin to explore in ways that I had never had an opportunity to do before. So that was an amazing experience, and certainly not only attracted me more deeply to understanding science as well as science in medicine, but allowed me to think about how I could approach his strategies for really doing a medical career that was founded in science. And he was a geneticist, and that certainly made one think about using genetics in some way.
Were there any early patients that you encountered that sort of shaped your approach at the same, whether during your residency or later on when you'd established your own lab, that changed your approach to science?
Again, very much so. One of the earliest patients that I took care of was a young woman who actually had a cardiomyopathy and was hospitalized because she was having recurrent ventricular arrhythmias, syncopal episodes, and was admitted to be given some new medications. In fact, the medication that she was hospitalized was one of the early trials for the use of amiodarone, a very potent antiarrhythmic medication that we still use today. Her course was remarkable only because she was so profoundly affected by this cardiomyopathy, but because she was so young. She was just like me. Except that there was something that propelled her heart to undergo this very horrific change in structure and fibrosis and triggering all these arrhythmias. And I could not certainly ever forget her clinical course and her ultimate demise, which occurred in a very short period of time, over 6 months, when I had the privilege of taking care of her. And I always, somewhere in the back of my mind, wanted to know, why did this happen to her? What was the root cause of this disorder? She did not have coronary disease. She had no risk factors for bacterial infection, rheumatic heart disease, or any of the other known etiologies of profound cardiac remodeling. And I think that in the back of my mind, that stayed with me for a very long time. It took more than a decade to be able to think of strategies to address that, but she was certainly a very important motivating source of that discovery.
Atrial natriuretic peptide, the structure, the function, the sequence of it, it overwhelms the beginning part of your CV. What was that time like when you were on the hunt and when you were first working on ANF?
Well, the way I came to work on ANF was really rather unusual. I had been a cardiac fellow and doing research under the direction of Dr. Ed Haber, the late Dr. Ed Haber, and working in the laboratories of Dr. Charles Holmesy and Robert Graham. And those two wonderful scientists and mentors were devoted to trying to understand adrenergic receptors. So too was another giant, Dr. Robert Lefkowitz, who recently received the Nobel Prize for his fundamental and seminal discoveries of G-protein coupled receptors, including the adrenergic receptor systems. So we had been toiling away, I had been in particular toiling away without a whole lot of success when I came home on a Sunday afternoon and read an article in Science magazine. And that article described the protein analyses of a small molecule, about 21 amino acids, that encoded what appeared to be a diuretic that was made by the atria of the heart. So it was an intrinsic diuretic within the heart structure. Any cardiologist would be interested in that. And so my interest was really piqued. But in addition, and simultaneous with that, was the recognition that there were new technologies that had come into play to be able to take a little piece of information and to expand the knowledge base. And specifically what I mean by that is the person who was the senior author on that science paper was Phil Needleman. And he had used hundreds, if not thousands, of rodents to purify the peptide to get sufficient quantities to be able to deduce the amino acid sequence. Using molecular approaches, one could take that information and produce degenerate oligonucleotide probes and to screen a library from 1 or 2 rodent atria and to actually learn about the molecules that encoded that protein, the cDNA and subsequently the gene. So I had been floundering away trying to do this kind of science in the adrenergic system, but didn't have that critical one piece of information, the amino acid sequence. This provided an opportunity to jump shift, if you will, and to use the same technology with the seminal piece of information that I was lacking in the adrenergic system. And so I turned to working on ANF, and I guess the rest is history. It was a great and exciting time. It showed the power, in my mind, of molecular approaches in the cardiovascular arena, and it also changed the way I felt we could do science in understanding heart disease. Most cardiovascular research in those days was largely directed to understanding mechanics of heart function, vessel wall structure and abnormality, and directly jumping from that information to therapies. Those were the years of bypass surgery, ultimately percutaneous interventions and the like. And while those have been enormously fruitful in saving lives of hundreds of thousands of individuals, a lot of the work was predicated on a mechanical understanding of a blockage of a structure that needed repair and opening. What I was more interested in is why did something happen? What was the root cause of disease? And what was the root responses of the heart to all sorts of different triggers and stimuli that were thrown at it throughout life? I looked at ANF, an endogenous diuretic, as one potential avenue by which the heart could perhaps respond to high blood pressure. And my interest of it therefore was broadened and more deeply because it really provided me a handle in getting more deeply into the heart and the myocyte.
Do you still work on ANF at all?
Yes, very much so. ANF is one of the molecules that is reactivated and expressed, as well as the brain natriuretic peptides, BNP, in response to all sorts of cardiac pathologies, including the cardiomyopathies. Indeed, it's one of the most profound signals that is upregulated in expression at the protein and the mRNA level, and we follow it to understand when, in fact, there's been cardiac remodeling and hypertrophy and heart failure and the like. So yes, very, very much so, but more as a marker of another form of disease rather than interest in its actual physiologic and pathologic processes.
So your more recent work has been looking into mutations in various proteins genes that cause anything from heart failure to cardiomyopathy. How many has your lab found over the years?
Well, we don't keep a running scorecard. We know that all of the cardiomyopathies, and indeed the majority of congenital heart defects that have a genetic etiology, are profoundly heterogeneous. A very poetic fellow in my lab once said, there are many ways that the heart can fail, and he said that after having broken up with his third or fourth girlfriend at the time. So I think that I think that's really true, that there are many, many genes, but what we've been able to do, and perhaps more than the actual number is important, is these genes often function in a shared pathway. And that has provided us the opportunity to cluster genes and the hundreds of mutations in each of those individual genes in terms of insights that are perhaps more illuminary than just numbers of genes and numbers of mutations. That's a long-winded way of saying there are, scores of genes and hundreds and hundreds and hundreds of individual mutations.
Within those genes, right. Have any of them come as a surprise?
Oh, very much so, very much so. Early on, we assumed that one of the mechanisms by which a heart cell could increase in size, because they're largely terminally differentiated, so they don't replicate. One doesn't get hyperplasia of the heart. Instead, one gets an increase in the size of the heart. And when one was thinking about that from 30,000 feet, you might expect that genes that are involved in growth would be activated in the setting of those diseases. So in the field of cancer biology, there are a whole lot of genes that have been characterized that promote growth of cells. One might have thought that there could be activation of oncogenes that would cause heart cells to increase in size. I was shocked, is the only word to describe it, when we learned that a simple set of proteins involved in contraction could actually trigger the heart to grow in size. That remains an incredible mystery. We're making some progress in understanding why, but I don't think any of us thought that a contractile protein, what is basically a structural protein, would trigger growth of the myocyte and indeed of the entire heart.
Which contractile protein?
Well, there are now 8 really robust ones that have been identified by ourselves and many others. They include the myosin heavy chain, myosin binding protein C, actin, troponin, and the molecules that make up the sarcomere, the contractile apparatus of the myocyte.
In order to find some of these mutations, you have relied on finding large pedigrees or families wherein, or some of the time, you've relied on finding these families and getting them together through picnics and family reunions. Now, how much of this starts with that, with trying to find the needles in the haystack and gathering all of them in order to study them? How open are some of these families to being studied?
Families that have a condition that profoundly impacts their health or indeed their survival are amazingly receptive and are great advocates of getting scientists and physicians involved in understanding the causes of their disease with, I'm unequivocally certain, the idea that this will promote discovery of cures. We don't often get to the cure stage, but we can't get there, in my opinion, without knowing the root cause. And these families have been amazingly helpful and endorsing of this work. We actually have never put on the family reunions or the picnics. We simply have said, if we came and saw you and some other family members, do you think other relatives might be interested in coming as well? And that has triggered them to orchestrate these amazing events that have been profoundly informative because in that scenario one can see the full spectrum of clinical manifestations from young people who we will subsequently learn might carry a gene defect and have no manifestation, all the way through to the individuals who are profoundly affected by it. So it's an amazing opportunity in a very short amount of time to see a spectrum of disease that could take decades to learn about if one saw patient by patient by patient.
How do some of these families, or even specific patients that you've worked with, react when you tell them that you have found the mutation that might lead to an explanation for what it is that's happening to them? I imagine there must be a psychology component to what you do.
There is. We call it psychocardiology. Genetics has always been deeply associated with the recognition that if you discover a genetic variant that is inherited, and most of these are dominant, which means that an individual who carries a variant, a mutation, will have a 50% chance of having all of their first-degree family members, their parents, their siblings, and their offspring also have that same mutation. So when one makes that discovery, you have already classified a family into affected and unaffected in very definitive ways. Early in the years of genetics, we thought that that was prophecy and definitive evidence that someone would get disease. Now we know that's a more nuanced likelihood, because many other genes in our body and our lifestyles and environmental factors influence the expression of those genes. That being said, many of these are very profoundly impactful in terms of an individual's human health. Individuals who receive the information who are affected are not surprised because they know they have clinical disease. And their initial response is often, how do we fix it? Mm-hmm. It takes a little time to recognize, even for an individual who is affected with a condition and has had it for many years, that this also implies they may give it to their offspring. And that's a very hard and difficult discussion that takes time for people to recognize and understand. And we've seen some families who accept that and who realize that they had no opportunity to prevent that transmission, that this was life, and other families who have had just devastating responses to it. We try to support these families in many different ways, which are not about cardiology, but there is no question that the responses are probably highly variable based on the strength of that individual family. Over time, I would say the vast majority of people who learn that they either are affected or are not affected are really glad that they've chosen, and it is their choice to know that information. For individuals who know they've passed it on to their offspring, their first response is not always how can we prevent the disease, but what are the things that we can modify in terms of their opportunities to change lifestyles and the like that might benefit their long-term survival. One of the good things about studying families is that often you'll find that there is an elder person in the family, a grandparent, a great aunt, or someone who has lived a long and fulfilled life even though they have the disease. And it's those individuals that you can point to when speaking with a family member who now knows their child will go on to develop the disease with high likelihood that really helps matters. The other thing is cardiology has enormous opportunities to prevent some of the most devastating causes of intrinsic heart disease. AICDs have revolutionized our ability to prevent sudden death. The development of antiarrhythmics to also suppress ventricular irritability is key. And of course, cardiac transplantation is ultimately gene therapy. We don't like to have to go down that track, but there are lots of opportunities to prevent some of the really worst and devastating consequences of these disorders.
Given these complexities, why not just work in mice? I mean, you do have several papers where you've explored some of the mechanistic details of things that you found in humans within mice, but that seems to be a heavy burden to also carry.
Well, I think first and foremost, I am a physician. I am not a mouse doctor, although I do have several of those small four-legged patients as well. And my motivation for science still remains deeply entrenched in my interest in improving the lives of people around me and improving their ability to have healthy, productive, and as long and happy a life as is possible. The mice are another mechanism to explore things that we can't do in people. The mice provide opportunities for us to remove a heart, to study different signals that are activated by the heart that we simply cannot get from our human patients. So I see the mice as similar to looking at DNA from a patient. It's a part of the process that ultimately we really want to drive back to human health.
You've also got some citations on your CV where you look at hearing and ear malformations and otitis media.
Yes.
So the ear's not necessarily next to the heart. So what brought up that interest?
Genetics will provide you continuing medical education credits in all sorts of human disease. When you see a family who you went and recruited to a study because they have a disorder that you are interested in, you can't help when you clinically evaluate them to notice that the individuals with heart disease might also have something else. And sometimes that something else is completely out of the blue, including a malformation of the pinna, the outer ear of the heart, of the ear, or that they're all wearing hearing aids. And so one of the families that we saw quite a few years ago, actually a family from Michigan, had a progressive dysfunction of their heart, dilated cardiomyopathy with a profound increase in the incidence of heart failure that was inherited. And individuals who had this disorder for many years all had sensory neurohearing loss. And when we ultimately discovered that there was a transcriptional coactivator that was involved in causing the pathology, we quickly realized from work by others that this same molecule was involved in hearing loss. And so this is a hand and heart disorder. And ultimately that's telling us something either about developmental biology or about shared molecular processes that different cells and tissues use when they provide differentiation and functional generation of the cells that are necessary for that organ. I find that some of the most fun part of the work that we do because you don't know what you're going to discover and you don't know what you're going to have to learn. So it makes you go back and relearn about the bones of the inner ear, the molecules that are involved in hearing, and it's a very creative way of thinking about it. it also does something very, very important. It makes you collaborate with others. And that has been a personal joy, and it also allows one to think about things in different ways, because we all come to a problem with a different group of background, pieces of information, and approaches. And we can learn a lot from our colleagues by collaboration.
So speaking of your colleagues, you landed at Harvard fairly early, but you've stayed for your whole career. What has that environment provided to you that has kept it so tantalizing? Because I must have heard your name in conjunction with every different chairman of medicine job across the country.
Well, you're very kind. I doubt that's the case. I went to Harvard as an undergraduate, and I came back as a faculty member years later, not with all honesty because of the name Harvard, but because of some personal issues. As you well know, I am part of a duo, and when I had been in my internship and residency training, my husband and scientific partner was at the NIH. And so we had this 50-mile commute each day in each direction that made life more complicated than need be. When he was recruited to Harvard, it was an opportunity for us to consolidate our lives into the same city. That's been good. And that's also something that's probably retained us at the same city, in the same city and at the same institution. That's a personal reason, but the far more important reason is the joy that we have had in really having tremendous individuals come and work with us at that institution. The medical students, the postdoctoral fellows, the individuals who are in the Harvard, MIT, HST program and the like are smart, they're creative, and they really drive science in ways that makes it exciting for everybody around them. I'm sure those same individuals are at many other institutions, but the ones that we've had the privilege to work with have just really made us say, why leave?
Given your approach to mentoring, do you ever talk to your students about work-life balance? I mean, I know you have 3 children, as do I, and that can be a really difficult thing to juggle, especially at the tender age when you're having your children and facing writing your first grants and balancing the work in the lab, which can have no natural, normal hours.
Right. I spend a lot of time talking, in particular to people in those states of their life that seem as if there's no solution. And I think that one of the ideas I try to remind them is that having children and having a life outside of science is important, and it enriches your science. It will only make it better, I'm absolutely convinced of that, because it reminds you about why you want to understand causes of human disease, so that kids like your own can grow up healthy and happy. So that's an important ideal, part one. Part two, I think that as there are many ways to approach a scientific problems, there are many ways to approach getting through the time management issues. Some of them require just plain money to throw at it, babysitters, weekend care when you need to write that grant, or perhaps recruitment of an aunt, a family member, a mother, etc., who can participate. And if those are the ways that work, and the hours that you work are at home or in the middle of the night or early in the morning, it doesn't matter. And as long as I think people recognize that the system allows for that flexibility, that they hopefully won't give up. Because most people who have a background in medicine or in basic science are certainly smart enough to figure out how to manage their time. I think that it's a pent-up problem that often is constrained because patient care does require being there. And so then when am I going to have time to also take care of my own family and do this research? And it may mean that you need to back off on some of the things that are less flexible for a period of time. But it doesn't mean you have to give them up entirely. And I think that just posing those ideas sometimes helps. Not everyone can do everything. Certainly not me. So I think that at times people also need to realize what are their personal priorities, and are they willing to miss school performances to spend more time in the lab on a day that really mattered for a professional reason or not. And ultimately, I think that's what makes us all a little bit different. And as long as I think the people that I speak with know that there is no right answer, it's only your choice and your decision, that I hope they feel more comfortable after talking about it.
Have you ever considered a different vocation?
What would I do if I was not a doctor? That's a very, very scary thought. I think I would still very much want to do something in science. I actually, a long, long time ago, wanted to understand how we could grow crops that would feed people in places where fertilizer and all sorts of access to the kinds of things that happen in agriculture in the United States were unavailable. And I actually worked on projects to provide nitrogen fixation in rhizobium plants to allow them to have intrinsic fertilizer. So I love to garden, and perhaps I would go and do something related to a more global issue. I think there's incredible amounts of science that have exploded in those arenas. Same too with alternative energy approaches and the like. So I think there would be something science-y about what I would do. I would hope it might take me outside a bit more. I'm not someone who likes to be inside all the time. So those would be really interesting things. And the other thing I probably would do would be to teach. I would probably do some teaching.
Well, I think I can speak for the bulk of the patients who've ever seen you and the rest of us in science who've watched your work that we're delighted that you did choose this career.
Well, thank you.
Well, thank you for joining us today, Cricket, and we
Pleasure.
appreciate hearing your story.
Okay, thank you.